Hi everyone, good afternoon. I'm so happy you could join us for the special lunch hour lecture. My name is Rosemary Eldridge. I'm the Director of Communications and Programs here at the Catholic Information Center. And on behalf of the CIC and our Director, Father Charles Chulos, it's my pleasure to introduce Father S. Joseph Tam for today's lecture on the ethics of CRISPR and genetically modified humans. Father Tam was born in Hong Kong and moved to Canada when he was 15 years old. He currently resides in Italy. He holds a medical degree from the University of Toronto alongside several other degrees in mathematical sciences, philosophy, and theology. He practiced family medicine for several years before entering the seminary and was ordained a priest in 2004. His doctoral dissertation, which he received with high honors, was on the secularization of bioethics, a critical history. Father Tam is former dean of the School of Bioethics in Regina Apostolorum, where he presently teaches bioethics. He is a fellow of the United Nations Educational, Scientific, and Cultural Organization, Chair in Bioethics and Human Rights. He is also the author and editor of numerous articles and books, including The Secularization of Bioethics, Religious Perspectives on Human Vulnerability in Bioethics, and Interreligious Perspectives on Mind, Genes, and Self, among many others. We are so happy to welcome not only Father Tam to the United States and to Washington, D.C., but to our CIC community as a whole. And with that, please join me in welcoming Father Tam.
Thank you. Thank you, Rosemary, and CIC for inviting me to give this talk. I guess about several weeks ago, almost a month ago, I was invited, and I gave them two options to talk about something on bioethics. I said I could be on CRISPR or on ethics of coronavirus, and they chose CRISPR. I don't know why. I thought coronavirus is much more a hot topic today. But anyway, this is something that I've been involved in. I mean, the ethics of this the last several years. I was also, I guess, present in Hong Kong when the news broke, when the Chinese scientist said that he made these two genetically modified babies. So I had kind of like a personal interest in this topic for various reasons. This is more a slide going back to grade 12 biology for those who might not remember. Genetics basically has to do with the fact that most of you might, I probably won't go too much into it, has to do with the fact that we are all the cells are made up of chromosomes and genes. And the strange thing is this: if all our cells have the same DNA, how come we are not just one big blob of cells which are identical, right? So we have all different cells in our bodies, and they express differently. No? And that's the question: how is this possible? So basically, this is the in some way a secret. How come certain cells are expressed differently in the body than other ones? And the secret there is basically how the genetic code somehow gets translated and expressed, in different cells through different processes, by which these expressions allows for differentiation. Okay, so the cells are different. Okay, and so this is, I guess this is grade 12 biology, just for you to remember. But the main thing is actually the fact that the genes are expressed through a translation into different proteins, amino acids. And these expressions are carried out, and then the building blocks, of the cells, the tissues, and the organs, and so on. Okay, now we talk about what we call genetic engineering. Remember, genetic engineering is not something totally new. We've been doing this for ages. Okay, obviously, in those days, it's much more primitive. It's through selective breedings, and by breeding, you know that you can push a species or certain type of vegetables or fruits or even animals to go in a certain direction. Okay, and we've done that, manipulated genetically different species through the ages. But recently, in the last, I guess, 30, 40 years, we've been able to successfully do it in a much more powerful way, effective way. Okay, this is another important, I guess, idea about genetic engineering. It has nothing to do with genetic engineering, it has to do with viral infections. We're not talking about the coronavirus, but it's important to know that one of the problems with genetic engineering is not changing the genes, but how do you target the parts of the genes that you want to change into the area, okay, that you want. Okay, it's the transport system mechanism. Okay, so we might be able to change the genes, but how do you make that change so specific that it goes to where you want it to go? Okay, that's the problem. Here, we know that it happens naturally, okay, in infections, viral infections. When the virus attacks you, or any cells, it inserts a certain information, new genetic information, into the cells. Okay, and this information is very important because this is how, to a great extent, we can make genetic engineering possible. Okay, we need to find this vector, they call it vector. Virus is not the only vector, but it's the most used vector to make this change possible in the target cells you want it to happen. Okay. So genetic engineering has been going on since the 70s, 80s. At first, mostly on plants and animals for agriculture and food production. Okay, this has been going on. You've heard of GMO and the whole debates about that. I won't go into that today because this is about human genetic engineering and not about animals. But basically, GMO is basically the ability to splice certain genes from a different species into another species. Right? Here we have the example of a fluorescent rabbit. Rabbits aren't usually fluorescent, but fishes are. So you can, okay, splice a fluorescent gene from a fish into a rabbit, and they've done that, and it's possible. So this has been going on since the 70s and 80s. So what is the advances? Many advances, obviously, in this field of genetics. You know, you've heard of the Human Genome Project, right? So this is different sciences come together. Right? I talked about breeding. I talked about the viral infections as a vector. I talked about GMO. Now this is another piece of the puzzle, which is the Human Genome Project. Okay, this basically completed in the year 2000, symbolically. It's basically to map out the genetic map of the human person, okay, fully. Because in order to say I want to change the genes of something, I need to know what I want to change and what that change will do. Where is it happening, right? So unless I have a map, I can't really say this is where I want to make the change and what that modification is going to have, what effect that modification is going to have on the cell and also on the animal itself. So here, this is a project that started as basically in the 90s and finished in the year 2000 symbolically. And they found that the human genome is about 3.2 gigabytes. So it's not that big, right? It's not that big. Now, gene therapy. Going back a little bit before that, this has always been a dream of the scientists since they discovered the DNA. And remember, DNA was discovered shortly after the Second World War. Okay, and they said, can we, okay, modify the genetic information of human beings? Mostly, I mean, at that time they were thinking about genetic diseases. We know that there are a lot of people with hereditary illnesses, and it's quite miserable. And they were thinking maybe if we could correct the genetic defects, then these people would be cured, right? So this is a very attractive project since the 70s again, with all these other technology coming, converging. So they've done that, but then it wasn't until 2012 that we have what we call CRISPR, which is today's talk. So they have different methodologies trying to do the same thing. It's not that easy, okay, to change the genetic information. First of all, they didn't have the map, right? Second of all, they don't know which part of the gene is responsible for what, right? And then the question about reaching there, okay, so the vector problem, and then making a change so specific that they don't want it to change somewhere else, because it could be disastrous if I change, okay, what I wanted, but also made a mess of this genetic code somewhere else. So it has to be so specific that the change is targeted in the place I want and would have the effect that I want, right? So there's some histories of difficulties here. We have a very famous case of Jesse Gelsinger, who basically died from some experiments because it was not done properly, and science basically had this setback. CRISPR, okay, stands for this: clustered, regularly interspaced, short palindromic repeats. A mouthful, okay? What is CRISPR? What is interesting about CRISPR, okay, is that it was discovered less than 10 years ago, okay, in Berkeley. What the scientists were doing, actually, they were not looking for a tool for genetic engineering. They were studying the defense mechanism of bacteria cells. They found that bacteria actually has a natural defense system against viral infections. Remember, I was telling you, the virus can infect a cell and change the DNA, okay, or the RNA of the cells, be they bacteria cells or plants or human cells, okay? Then over the years, evolution has given these cells a way to defend against viral infection. And then they found out that one of these defense mechanisms is the ability to recognize the virus DNA. Because once you get infected once, you recognize what the pattern of the DNA or RNA of the virus is. And then you can actually, this defense mechanism is very simple. You can go and attack it and cut it. Okay, once you cut it, it becomes inactive. Okay, and so it's a defense mechanism. The bacteria is able to defend against the virus when it attacks it the second time. Since it recognized the first time this virus has this DNA configuration, the second time it attacks it, it recognizes it and has this CRISPR type of protein inside its cells, and it could recognize and cut the virus when it attacks it the second time. Okay, so this is what CRISPR actually does. There's a video. Can you click on the first video? Okay, that explains a little bit about how CRISPR works. The first one, not the second. Okay.
Oh, there's two.
There are two, yeah. The first one, oh. The first one, yeah. The YouTube one. Yeah. Oops. Yeah, no sound.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing wheel. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Okay, so basically, the amazing thing about CRISPR is that they found out a way. I remember I was telling you that you have to be very precise in the way you target the genetic information outside of this 3.2 gigabytes of information. You have to pick out like one base pair, right? That's quite difficult, you know? But they were able to do that. Okay, it's just kind of like what we call if you have used Microsoft, right? That you have a whole book, and you just want to say, okay, there's one word I want to change in this whole book. Okay, so you can do a search and replace of this one word. Okay, a computer can do that very fast. You know, you just type in the word, matches it, and then it goes there. You can replace it or you can delete it, whatever you want with it. This is exactly what we could do now with any genetic information, in the sense that any cells that has DNA, and you know the sequence, and you know where you want to go, and you want to go there and do something to it, you can do it with CRISPR. So CRISPR can basically do one of several things. It can cut it out, it can suppress it, it can potentiate it, it can make it more active, okay? So for example, you know that in this gene of this cell is used to produce insulin, you can actually not just suppress it, but to make it more active so that it produce insulin, for example. So this is a very powerful tool that they discovered less than 10 years ago. So as you can see, as a result of this, there is what we call a phenomenon where studies and interest in the scientific world on this new technology has grown exponentially. So basically every year, the number of studies on CRISPR has grown. Right? Basically back in 2012, when you first discovered the thing, there were like less than 500 studies. But now, in less than 10 years or 5-6 years, about 4,500. Probably this year, 2019, you will have like 10,000 publications on this one technology. Obviously, this is not only on potential for curing diseases. There's a lot of applications in plants and animals, okay? And we'll just briefly touch on that. And then we look at how this could be applied to human beings, okay? Either people like you and I, adults, or embryos, if we could do that. Okay, remember, it can be done. The question is whether we should do it or not, okay? So plants and animals, what can you do? Okay, you can make, for example, the pigs more muscular, so, okay, you can have more meat. You can have pigs really miniature, so that you can keep them as pets. You can have a cross between a chili and tomatoes, so you have spicy tomatoes, so don't even have to mix them together. Or you can basically make the mosquitoes deformed so that they no longer could reproduce, okay? So there are many, many possibilities when you can actually genetically modify any species, any cells you want, okay? So this is something that they've been talking about recently, about neutralizing mosquitoes of malaria, above all, or any disease-carrying mosquitoes. And they could basically wipe out enough mosquitoes that they will no longer be infectious or carry the malaria. Okay. Now, we can talk about how you do this for human beings. How do you apply this technology? Other than curing or correcting genetic defects. You can use that to also, for example, detect viruses or bacteria. So you can use it, to fight infections. Because remember, once you know the genetic code of these diseases, these bacteria or germs or whatever, you can target them. Cancer cells, you can either target the cancer cells or can potentiate what we call the immune system of the red blood cells so that they will fight the cancers and so on. So many possible applications, tons every year. Now, this is actually where we get to the talk, okay? What can we do with human beings right now, okay? So the possibility is there, the technique is there. Remember, this has been talked about since the year 60s, when bioethicists were thinking about possibility of genetic engineering of human beings, and they're saying, what can we do and what should we do if this technology one day becomes a reality? And now it has become a reality, okay? So this is upon us today. It's no longer science fiction. So here we can see that there are two types of cells, okay? We talk about adult cells, we call somatic cells, meaning people like you and I, okay? People who are born already with the genetic information set in stones, you can change that. But with the CRISPR, you can change that, okay? Or we talk about germ cells, we talk about embryos, sperms, or eggs, okay? So there are two categories. And then, for what reason, you can talk about therapy, meaning that, okay, if you have a defect, okay, in your system, the DNA you have genetic disease, whatever, okay, you can treat the disease or you can cancer or whatever, the things I told you about. You can use this technology to treat, okay, any illness in either the adults or the germ cells, okay? Or the other possibility is enhancement. Okay, here, it's no longer just therapy, treating some illness, but potentiating, okay, increasing a certain power that doesn't occur naturally. Okay, so this is enhancement. So here basically you have four categories: therapy, enhancement, and somatic, or germ cells. Okay, so we have four categories more or less, and you can think about what can we do and what we shouldn't do. Okay, what do you think, the audience? Okay, usually in the first category, does this work? The red thing? No. Oh, this. Oh, it doesn't work. Laser? No. It's okay. Don't worry. So therapy of somatic cells is something basically. Let's say you have an illness, genetic illness, or use this technology for treatment of illnesses. Most people would say, in this category, is what? Okay, right? This is the easy one, okay? And this is also the easy one, okay? Basically, use genetic therapy, not for treatment, but for enhancement, okay? Most people would say they don't want this, okay? These two are much more complex. We'll get into that right now, okay? And see the reasons for and against. So this is basically treatment, okay, of adults, okay, with illnesses, genetic problems. We just want to genetically engineer the cells that are diseased, okay, by these techniques. And there are many illnesses that are being treated now. So these are just some examples. And actually there are active trials since a year ago on some of these illnesses, such as thalassemia, such as LCA10, which is an eye disease, such as muscular dystrophy. So there are actually trials ongoing, and then they're getting good results. And there are many other possibilities, okay? So here, I just mentioned, most people would say, yes, no problem. Now, difference between somatic and germline gene therapy is that somatic cells is basically adult cells, okay? And whereas germline cell, once you change the genetic makeup of those cells, the embryo, whatever, they will get passed on to their generation of future progenies. So it's the difference between somatic cells, because you change my gene, but then it stays with me. If I change the genetic information of a sperm or egg or embryo, okay, that stays forever. We know already how embryos come about through IVF, okay? And nowadays, they also can possibly do screening, because once you have created embryos, before you implant the embryos into the body, into the womb, it's possible, technically speaking, to do a certain kind of screening to say that I don't only want to implant embryos that are genetically healthy, okay, rather than those which could be genetically defective, right? Okay, so the question here is, should we, okay, use this technique to cure embryos with genetically defective genes, okay? And one consideration is that it has to go through, most of the time, IVF. It's possible to do it without IVF, but that's the current state of technology. You have to use IVF, which the church already prohibits, okay? On the other hand, like Francis Collins says, there's actually no need for that at the moment, because you can already screen out the genetically undesired traits in embryos, okay? So why would you want to actually use this technique to cure embryos of genetic disease, okay? So basically this is an image that you use, right? You have basically 3 cars, okay? One of them is defective, okay? Would you try to fix a car, okay? And so you have a brand new car which is basically not working well, but then you can fix it. Or you can actually just say, I picked the cars that are already perfectly running. You can do that already. So why would you go through the last type of treatment to get something that is, you don't know how this is going to work, and you don't know if this car in the long run is going to work properly, because you're making a lot of manipulation on this, right? So basically this is what Francis Collins is saying. There doesn't seem to be a strong enough reason to go this way when you can already create embryos and discard, okay, the genetically defective embryos, or not to implant them, other than finding specifically those embryos which have genetic disease and try to cure them with genetic therapy using CRISPR. Okay, this is what Francis Collins is trying to say. Obviously, from our position, creation of embryos through IVF already is problematic. Okay. So here, this is what we have said so far. Okay, for therapy of human germ cells, you need to use IVF, so problematic, and from a practical point of view, it's rarely necessary. Okay. Now we have to go to this part. Okay, we want to enhance embryos. Okay, and most people would say no. This is a study, a question that was asked. Okay, and people basically are in favor of treatment of embryos, but they're very much less in favor of enhancing human traits through genetic engineering. So why would we want to do this? Once again, there is definitely a certain pressure to create the perfect baby, right? So this is rather than be surprised, you don't know what this baby is going to be. You would, if your technology allows it, there's a certain business, okay, and a certain exploitation in the sense that you wanted to create a baby that is perfect. Okay, this actually is the title of a book. Okay, not really a title I make up. It's Superman, Wonder Woman, and Perfect Babies. But in general, most people will say at this stage, no. Okay, we don't want to create perfect babies through genetic engineering because it could sound scary. It could think about all the scenarios of this possible, but the technology is there. Technology is there. Okay, now there is a distinction that I want to make between enhancement and therapy. Well, I would say that therapy is basically if someone is, let's say, born short, okay, this is kind of like a normal curve, general population, not the height of everyone is kind of like a range, right? So those who are really short on one end and those who are really tall on the end, and there's kind of median in every population, okay, every race and so on. So if someone is born really short, treatment would be basically putting that person so that he will attain, okay, correction of height in this case, to a more or less a normalcy, we would say, an average height. This would be considered therapy. Whereas if you say that this person is already normally tall, I mean average height, you want to increase the height of the person, okay, that would be considered enhancement. This is simplifying it, but this is how I would define it, okay? So therapy is when someone is below normalcy in this curve, you return that person to normality, the median height, and enhancement is when that person is normal, but you want to increase that potential, okay? That could be done to embryos. Obviously, we said no, but also technology is there also to do it to people like you and me, right? Adults. Why would you want to do that? Okay, what can you do? In some way, it's much more complicated question there, okay, in this sense, because we always try to improve our health. Isn't that a type of enhancement? I wear glasses. Is that a type of enhancement, right, of some sort? Let's say if I could enhance my health so that I could live another 20 years more, is that a type of enhancement? Or if I could live up to 200 years of age, people might want that, right? So there is a little bit more reflection to be done on this area, okay? Why we want to do that and what is finality? Why do I want to live longer? Okay, what is the meaning of life? What is the goal of life? What's the goal of medicine? Those are more difficult questions to answer. And so I would say that it's not an absolute no, because we do that a lot of times in what we do with medical technology, but we have to ask the question, why do you want this? Is this a good reason for you to use this technology? And there are many other concerns, okay? All right, so in terms of ethics, okay, I would say these are the questions that are being asked, okay, in terms of ethics of genetically modified human beings. Difference between therapy and enhancement, we saw that a little bit. Question about access, okay, who can have access to this technology, who can use it? About safety, is this technology safe? Can it cause errors? Do we want to create monsters? I mean, that kind of question, right? Justice, who basically can have power to use this technology? Would it be justly distributed? Okay, would there be creation of different classes of people, those who have access of this technology and those who don't? right? Dignity for us is very important because it has to do with, human life, new human life in terms of the babies, but also dignity, also for adults. The question here for us is question of why do you want to do this? Is this going to make you a better person? Okay, is this going to create a type of super race? But then, is it worth it? Okay, what are we going to lose? Are we going to lose our humanity in that process? One question about CRISPR is that unlike the other gene therapy technologies, it is easy to use, highly accessible, and cheap. You can order a CRISPR kit on Amazon for $100. Okay? And so there are problems that are associated with CRISPR that are not present with the other previous genetic engineering therapy, because here we're talking about something that is so freely available and so easy to use that we run into a question here about hacking, right? People could use this, anyone can use this, and who can have access to this, because you don't really need a big budget, you don't need a big lab, and you can actually do a lot of experiments on your own. So this is a new issue that was not present previously. Before, you really need a lot of money and research and technique. Okay, the question about safety, once again, I won't go into it too much, but in every technology, there are always concerns of safety. How to make sure that this technology is applied correctly. Okay, when you change the genes of any cell, how to make sure that it doesn't go off target. Okay, that's the whole question is about that. Okay, would it be used, and it could have long-term effects. Okay, how do you make sure that it doesn't happen? Okay, there are different questions here. Justice here has to do with the fact that, okay, who have possibilities? Is it going to be people who are rich, or countries who are rich would have a greater advantage using this? Is there going to be kind of discrimination as a result of this? If you don't use this, okay, let's say in the case of doping is already happening, okay, an athlete that does not dope might lose out, might not be able to compete because everyone else is in that, okay, industry, in the sporting, in sport, that sport he or she is doing is doping. Okay, if I don't do this, I will not be able to compete at all. Okay, and this is happening in certain sports. And that question also is raised. I mean, isn't there a kind of discrimination for those who want to be, for principles or for reasons, for religious reasons, don't want to be involved in this technology? For some Catholics say that I cannot use this technology, okay, to create embryos, okay, that are enhanced, let's say, in the future. But if everybody else is doing it and they have greater IQ and smarter and disease-free, then I'm going to lose out. Is my baby going to be able to compete with them? Right? I mean, that's a lot of pressure there on parents, peer pressure. So here, this is something that we have to think about, okay, when we want to enhance the human race. What do we want? These are the qualities that people usually want: to live longer, to be healthier, to be stronger, to be faster. Okay, those are the main things, okay, but also the other things. No? So you use it to create soldiers. I mean, you see this in all the movies and things like that. What do they want? If you could change the human race, okay, what would you want in you? To live longer, to be better looking, to see further, to be able to have a greater intellect, higher IQ, memory, happier, Okay, the question here is also not just to create a Superman. Okay, here the problem is also that these are all physical attributes that potentially, using genetic therapy or engineering, you could change the characteristics that you want. But we may not, okay, create a Superman, but a supervillain. Okay, what if you create someone who lives longer, okay, who's healthier, never get sick, is strong and fast and really attractive, but it's actually a really terrible person, okay? And you won't be able to kill that person because he's so smart, so rich, so powerful. What happens then? Okay, this is another question that sometimes we need to ask. So here, there's a whole chapter here that I'm not going to go into. The ethics of technology. Technology can save us, really, or can it destroy us? Okay. Now we go to the next part, which is about what happened in China recently, a year and a half ago. Okay, about what this scientists did in China. Okay, here is basically the map of the laws against embryos experimentation until 2017 or 18, okay, around that time. There's some ban in some of the mostly Western countries, okay. In other countries, they are more ambiguous, okay. But the thing is, this all changed when China, that in 2018 or 19, 2018, right? This scientists, okay, He Jinkui, announced, okay, in this conference in Hong Kong. I'm there. You can see me. Taking a photo in the front. That he created 2 babies using CRISPR technology with certain characteristics. Okay, so basically this is a slide from him. Basically, what he did, okay, was that he inserted certain genetic modification, okay, into these 2 babies that will protect them against HIV infection, okay? It's not that the embryos have risk of HIV infection, but he wanted to offer them a certain protection against potential infection in the future. So this is called a CCR5 mutation that he created artificially in these 2 embryos. And then when it came out, okay, there were a big storm, a media storm, about what he did, okay. We know that from that experiment, he implanted 11 embryos. 2 were born, 1 more on the way back then, now it should be born already, okay. So this is what happened back in 2018. 19. Losing track. Okay, and it's interesting to observe this because there are many news. I'm sure you've read some of that. Basically, the 2-story line. Okay, most scientists, okay, came up saying that okay, this is unethical because the technology has not been proven yet. It's not safe, and he moved too fast. Okay, he's an anomaly. Okay, he's a rogue scientist because his experiments, his techniques are not mature enough. Also ethically problematic because he did not really inform, the the parents adequately. He's not a doctor. He can't really have this kind of relationship. And what's going to happen to the babies afterwards? So all these kind of ethical reflection about what he did, and very disappointed. It's actually very interesting to look at the news because I was there in Hong Kong when he came and spoke, and basically he's been labeled as someone who is either too naive or is someone who is unscrupulous, okay? But I think there is something deeper going on there, okay? That when you listen to him, I realize that he might be, I would say, ethically naive. He didn't really know what he was getting into, and he actually did not expect this kind of reaction from the scientific community. Actually, when you look at the news, the first day of this story, when it broke out in China, he was praised as a hero. Second day, they took it down, and then he was condemned, right? So I think also we have to understand he is a product, okay, of the scientific culture himself that looks for, to be able to become famous, you need to be able to make a breakthrough, okay? And he had no problems of doing this because he felt that he was doing a good thing, okay? Because he was helping these couples who have HIV positive and preventing the children from getting HIV in the future. Yes?
Didn't he, I heard through Dr. Holman that he actually grew up in a village in
Yes.
China that had a high HIV, incidence of HIV
Right.
in its population. And some of that was attributed to the richer culture actually somewhat preying upon the poorer culture for blood transfusions of all things because they thought it gave them youth and all these. So there was a propagation of the HIV virus. And so, to say that he was cold and callous or unscrupulous, he actually thought he was doing a good thing in a
Right.
way, which...
Right. Well, I still think that what he did was wrong, okay?
Right.
But I think he's also a product, okay, of this culture, either in China or also internationally, because he traveled to the States and made presentations, he wrote in Nature and so on. So he's not totally naive. What I'm trying to say is that the next slide would explain it, okay? He is looking at the scientific literature from 2015 to 2018-19, okay, before the, basically 2018. Basically, the scientific community, okay, when you look at the reports, okay, this is the National Academy of Science, National Academy of Medicine, National Academy of Technology, okay? And basically, they are proposing that eventually we will want to modify and enhance the embryos, okay? This is the reports of these councils saying that we're going in that direction. Right now, it's not mature. Eventually, we do it. So he explains, some of his things, he thought, why wait? Why do we have to wait? Why can't we just do it now? Because if it's a good thing, go ahead, do it. In some of his interviews, he compares himself to Edward, who is the first pioneer who started IVF, because he said he started it without all these things either. He didn't do the safety tests. He didn't have all the protocol. He just went ahead and did IVF. And right now, this is, he got a Nobel Prize for that, right? So he was saying that technology doesn't have to be fully proven to be successful, and he compared himself to that. So basically, he's saying that reading the scientific literature, the scientists are going in that direction. And in fact, when I was in Hong Kong, I have the sensation. The scientists there are saying the same thing. They're saying that we should experiment on the embryos. We should allow this technology to move ahead. They call it a translational pathway, okay? So the trials of germline editing of human embryos should proceed in an orderly fashion. What they are upset about him, the way I read it, I guess I could be cynical, is that he jumped the gun. The scientists saying that we want to make sure that this is done in a way that is progressive, but in a way that we could convince the public that this is an important thing, but we don't want to make it so precipitous. And then they will have this kind of backlash. We don't want that. So they're playing a game by which we want to go slowly, and then when we are ready, okay, the public will be ready. Okay, so they're upset with him because he was too impatient. He jumped the gun. And he jumped the gun because him reading, actually, properly the scientific community mindset is that we will do this, but not now. And he said, why not now? If it is good, go ahead, do it now. I mean, this is another thing that I would say is somehow not so much heard about in this whole debate, okay? How much responsibility of his action is a product of the scientific way of doing things, you know? The competitiveness, the cutthroat nature of scientific progress, and so on. So here, this is what a conclusion is, that we have to be careful about enhancement in adults. Most of the time we say no for now, but remember, this is from a Catholic point of view. But what I'm afraid is that this is where we're facing in the future, okay? Yes to therapy. The documents I showed you already, in some way, is saying that yes, we should also use treatment for germ cells, for embryos, because that's good. Okay, we can get rid of disease once and for all. Even though it's rare, I mean, you're talking about very few cases, but it doesn't matter. These parents might want to have the embryos corrected. Yes, for human enhancement, because there's a very high, I guess, pressure, peer pressure I told you about, that's in sporting. Once you start prolonging life, and everyone is living longer and happier, would you not want to have that? If you have the ability to buy another 10 years of your life, would you pay for it? Would you? Yes? No? Yes? Most people would say yes. No? This is the way businesses run. So this yes, okay, this yes, and then moving down this way. Okay, even embryos, if you can do that for your kids, okay, that they would be smarter, healthier, more perfect. Why not? So this is what I'm thinking is going to happen in the future. To end this discussion, there's another point that has to do with another issue that after this news broke out with the Chinese scientists, there's a lot more reflection about who should decide, who should make these kind of decisions, whether we should put a stop to genetic engineering in embryos or not, okay? Here, basically, what I've noticed is that on one hand, the scientists are saying that we shouldn't move ahead so quickly, okay? It's not to be excluded, okay? But this is in the future. We want to make sure that there's broad societal consensus before we move ahead. But on the other hand, okay, when you read these two authors, they will say that actually they're playing a media game. The scientists really want this. They really want to be able to have the technology and decide among themselves. So there is a kind of like a thing going on there. And basically, you would say, in this book, especially that I've read recently, basically she's saying that the problem today is the fact that scientists the scientific community believe in self-regulation. Okay, we decide among ourselves what pace we want to go and what research we could do. Okay, and we don't want too much interference from the government. We don't want, especially from ethicists or the church, tell what they say that we should do. Okay, they don't like that at all. Okay, so broad societal consensus, is it a real thing? Okay, where basically the society, and not just the scientists, but people like you and me, have a say as to whether we want to go ahead or not. Okay, and what are the debates and how is this going to carry out? It's something that is still in the air. Okay, but right now they're very resistant to a type of open debate, because they say, well, usually the scientists say, this is too technical, leave it to us. Okay, the public would not understand this. The policymakers, they don't understand this. So we're living in Washington, I'm sure you come across this as well. So this is the last slide. So going back to my question, what is it that we want? The perfect human being. What is the perfect human being? So on one hand, the science might be able to give us a type of perfect humanity, but only in the physical sense, right? But then what our faith, our religion has been telling us is that we do want to create a new man. But this new man comes through a spiritual change. And ultimately, that is what really matters. That's true transformation, okay? Not a physical transformation, but something much more greater. And that's it. There's some references at the end for you too.
Unfortunately, we don't have any time for questions here in this space right here, but I'm sure Father Tam would be more than welcome to answer some questions right out here in the bookstore area. Thank you again so much for speaking on this important topic here at the CIC, and for all of you here in person for coming, and for those who are watching online. Thank you, and we hope you have a wonderful rest of the day.
Thank you.